my pleasure having you here in this small room. I guess that this morning we will be devoted to microcirculation, and glad that not only Professor Lee but also Professor Solano and Professor Chismar came this morning. Hopefully they can also participate in the discussion. And as usually with a small number of people, we can just go slowly and please feel free to ask questions and interrupt the talks, because I guess that for small tutorials it's always important having contact with the audience and having opportunity to ask questions and answer questions as well. So when I started this slide collection, I checked the internet and wanted to know what happened with microcirculation and laser during the last 30 years. So this when I put into the search engine laser and microcirculation, it is the number of the publications published during the last 35 years. It starts in early 80s and it goes up to 2015. You can see that the number of the publications which are related to laser and microcirculation were pretty low until 1990, and this is a landmark when in this year's the first laser apparatus were put on the market for commercial or research purposes. And since then, about 120, 130 papers are published each year related to lasers and microcirculation. If you make a more refined search, when you put laser Doppler and microcirculation, you see that almost 80% of the papers published during the last 20-30 years are related to laser Doppler and microcirculation. So the majority of the papers published during these years are using the technology I am talking about details today, and hopefully Professor Lee will also talk on these aspects as well. Of course, the science can be measured with many tools and many means. One is the number of the publications. Other specific approach could be how many citations are on specific areas. And this graph shows that during the last 20-30 years, the number of the citations are growing, and last year about 4,500 papers cited articles related to lasers and microcirculation. So therefore we can state that, okay, during the last 30 years, 3 decades, laser physics and microcirculation got tied to each other, and many investigations are done using this technology. I checked which was the most cited paper. There are several papers which are related to tumors, and in this respect, not the technology was important, but the biological meaning. But one of the most cited paper is this dynamic imaging of cerebral blood flow using laser speckle, which is a paper by Dan, Bolle, Mike Moskowitz, and Boas. Today we will talk about this approach as well. So let's go into details. What does does it mean microcirculation, and why is it important for life sciences, and what shall we know about and we like to know what's going on with microcirculation lasers. Of course, the circulatory system is essential for all creatures in the planet, and the circulatory system is one of the major attributes of life. Well, you'd like to explain what does it mean and how it is related to life. Just to have an example, if you'd like to explain how is a city working, big city, and there is one aspect to check. These are the roads, the traffic on the roads, the tubing system, and if you just observe these main roads and the tubing system, there is a way to calculate how the city works. Of course, this is not an exact description of a city life, because in the city, of course, there are many other aspects. But a major attribute could be characterized by the circulatory system and the microcirculatory system as well. So if we are talking about the human microcirculation or the human circulatory system, we have to state that the microcirculatory system is now with this terminology include capillaries and lymphatic vessels. Today I will mainly talk about the capillaries, neglect the lymphatic system, but it is still a very important way of or aspect to study the circulatory system, because, for example, the tumor progression is always tied to the lymphatic system, and it was regarded for decades that the brain doesn't have any lymphatic system. Last month I found a paper which is again emphasizing that even in the brain you can find lymphatic vessels helping to drainage of the plasma and the other constituents of the brain fluids system. So the lymphatic, the capillary system contains about 5% of the entire blood volume, which could be 250 milliliters of blood, which is a certain amount, and in this capillary system the blood is moving with a slow speed. In the big arteries, the speed of the individual red blood cells could in the range of 40-50 centimeters per minute. In the microcirculatory system, the speed of the individual red blood cells, few microliters per second, or maybe 10, 20 microliters. So this is also a very important issue, is that in the circulatory system there is an uneven distribution of the speed of or the velocity of the corpuscular elements, of the cellular elements. So therefore, even this question, what is the speed in the microcirculatory system, is very difficult to answer, because you go deeper and deeper, you see that there is a huge heterogeneity in the movement of the red blood cells, and especially if it is in a disease state. And even if you consider that, okay, the red blood cells move uniformly, but the white blood cells don't. So the microcirculatory system takes part in the exchange of nutrients, gases, and waste products between the body and tissues. So that it is also sometimes neglected that we study many transport processes in the living systems, but one of the major transport processes is the diffusion. For the diffusion, the system needs surface and time. So the diffusion is a slow process, even, of course, it is depending on the time scaling. But for the microcirculatory system, we have to always consider that it affects a huge volume and huge surface area, because the microcirculatory system is working in the major transport processes on the way of diffusion. So there are about, so it is always a question why people count capillaries, why people do quantitative measurements in a living system, but I guess it is also important to emphasize that we have to somehow describe the system, and mathematics and numbers and equations are helping to describe the nature of these systems. But we can also state that there are about 10 billion capillaries in the human body with a surface area of 500, 700 square meters. It means a huge area which provides a surface for diffusion. Diffusion for oxygen, diffusion for carbon dioxide. And also, one has to also consider that the heat transport is also essential. In the combustion, in the metabolic process, a lot of heat is produced, and a heat equilibrium is also achieved with the proper circulatory and microcirculatory system. So, of course, as we go deeper and deeper, we can also see how the circulatory system is constructed. It comes from big vessels, and there are branching, and this branching is also not a random process. A lot of people spending time to measure how many branching are, how the system is optimized. We have to consider that all these areas which are covered by capillaries and small venules are somehow developed to provide a proper and optimal exchange for nutrients, for gases, and also for heat. But if they are too dense, the system, it is killing the major cells. If the system is too separate, too big distances are, therefore the transport process, especially the diffusion processes, are at the limit. So therefore, even the microcirculatory system has a so-called optimization, and one approach is in the tumor therapy to kill the microcirculatory system, to hinder, to neglect the transport processes, and therefore the tumor cells can be neglected or can be prevented from resources. I don't go into details, but not all the microvessels are always open. There are microvessels which are closed and opened. There are regulated systems which are helping to close and open these systems. These are the so-called precapillary sphincters. These precapillary sphincters, like small taps, for certain commands they are opening and they are closing, and this is called recruitment of the microcirculation. And these precapillary sphincters are regulated by various processes, but mainly tissue-borne chemicals are helping to control these valves. Of course, there are central commands like the central nervous system or the endocrine system, which is helping to open and close this regulatory system. So if we are checking and measuring the details, we'd like to know how this system is regulated, how the blood movement is regulated within these capillaries, and what are the major rules for working in such a complex system. So when one considers that which tissues were first targeted to check the microcirculation by lasers, of course it is very obvious that the tissues which are superficial, mainly the skin, we get a lot of light from our surrounding. There is an interaction between the human tissue and the light, and of course light is absorbed, light is reflected. We can consider what's going on in the microcirculatory system. Today I won't talk too much about the microcirculation of the skin. I will consider another very important organ, which is the brain, which gets about 750 milliliter blood within a minute. It is about 15% or 14% of the cardiac output. Cardiac output means that how much blood is pumped out from the heart within 1 minute. It's about 5-6 liters depending on the body mass of the person, and also it depends on how big is the physical and emotional activity. But one feature is for the cerebral circulation that about 750 milliliter blood is provided to the brain, and this blood should be very well oxygenated, because about 20% of the total oxygen consumption of the body goes to the brain. So just consider our brain's weight is about 2-3% of the total body mass, and it is getting about 20% of the oxygen. And as we know, well, the brain is very vulnerable to the oxygen and blood supply. Few minutes or few seconds are enough to lose our consciousness, and it means that there are not too much reserve capacity within the brain tissue. There are no oxygen storage, oxygen pools. There are no pools for glucose. So therefore, it is an ongoing supply system which is required to maintain the brain alive. So the last 20-30 years of the brain research is focusing on the relationships between neuronal activity and blood flow, because it's well known that if the neuronal activity is high, it requires high blood flow, and if there is a disturbance between the coupling of the blood flow to the neuronal activity, it could be dangerous. The biggest danger is the so-called brain accident, which is also sometimes called stroke. It affects about 7-8 million people all over the world. This is now the third cause of the death, and second or first cause of the long-lasting disabilities. And as the society is getting older and older, we have to face another challenge. This is the dementia. It is very often related to lack of the appropriate blood supply of the brain. So therefore, this is not a question why it's important to study the cerebral circulation, and why it's important to know more and more about the details of the cerebral blood flow. So, how can we study microcirculation? Of course, there was a huge methodological armamentum before the lasers, because we can just do microscopy. Expose a tissue. In this case, the tissue is the this is the urinary bladder, and make a simple microscopic approach. In this case, white light illuminates the tissue, and of course some light is reflected, some light is absorbed, and we can analyze the contrast between the different components in the picture. Which is red could be regarded that this is the blood, and the borders are the borders of the blood vessels. If we do some dye or some trick, for example, we use the fluorescence, we can get other type of information. When it is a more complicated organ, for example the lung, we can check also some details, but as you see, not at least for an unexperienced eye, it doesn't tell too much. A lot of studies has been done in a very simple model of the vascular development. This is a chicken development. If you just open an egg, you can observe the blood vessels even with naked eye, and if you use fluorescence microscopy, you can check details. What are these details in this case? Number of the vessels, the size of the vessels, the diameters, and even it says a lot for people what are the angles and how the new vessels are developed and how is the dynamics of this vascularization. It can be also done in more sophisticated way. This is a small chamber. This chamber is exposing the skin, and under the skin you can see microvessels, and this chamber could be filled with various substances which are affecting the microcirculation, and you can see details even with microscopy. What is lacking on these methods? First of all, one can regard that there is no microcirculation between the big vessels. There is. Just you cannot see them because you cannot penetrate deeper and you don't see. So therefore, sophisticated methods like polarization microscopy or other techniques were used to get deeper and see deeper structures. And even now the microscopy is a pioneering word of approach of the microcirculation. Now most of the microscopic approaches are using multi or just 2 photons for getting deeper and getting more detailed approaches. And this picture shows you that even now it is very important to investigate the dynamics of vessel formation, how many vessels are produced within a certain amount of time, because as we started, if the blood vessels are occluded, you can kill the tumor. If the blood vessels are developing very quickly, for example, in a diseased eye, sooner or later the patient will lose the vision. So therefore, getting information about the number and size and structure of the blood vessels is important. In this detail, we can check also how useful would be an anti-tumor development, but I don't go into details. But with the microscopy, we can check white blood cells and other structures. Intravitreal microscopy is even now used, and more microscopes are developed using laser lights, because I will tell why laser light is important. This is an exposure at small vessel, and we can also detect small blood cells like white blood cells penetrating and So, of course, there are many approaches for measuring the microcirculation. Most of or almost all the approaches are indirect approaches. Indirect approach means that we measure something and calculate something because we regard that there is a very good correlation between the observed variable and the real variable. This sometimes it's true, sometimes it's also biased, but we measure with indifferent gas methods, hydrogen clearance, isotope methods, many methods. Even now we use positron emission tomography for checking microcirculatory networks. So, what are the principles of laser Doppler flowmetry, which I mentioned, producing even now the most number of the publications a year? This is a method to assess the tissue microvascular perfusion. A laser beam is used. Laser beam is used because it can be focused well. Laser beam is used because laser, you learned during the last days, is a monochromatic or producing monochromatic light, hopefully coherent light, and for diagnostic purposes we always use soft lasers. It means that we expect that there is not too much interaction between the tissue and the light. It means that the observation is not affecting the area under observation, so therefore we are not biasing the system. In the laser Doppler flowmetry, we use a light source, which is mainly a laser light in the range of 540 nanometers till it can go up to the infrared area, which can be around 780, 800 nanometers, depending on the type of the laser used. And in this wavelength, the penetration is couple millimeters. It means that most of the light is backscattered, and of course huge part of the light is backscattered from static compartments, but some of the light, some of the photons are interacting with moving particles. In the blood, the most abundant moving particle is the red blood cell, and therefore if it hits the laser light, if it's hitting moving particles, it is reflected, and if we are lucky enough, we can collect the reflected light, and the reflected light is containing the properties of the moving red blood cells as well, and we'd like to analyze these properties, which could be the number of them and the velocity or the velocity distribution. So therefore, there is a sophisticated or not sophisticated for physicists or for engineers. It could be sophisticated for biologists or for physicians methods when we go from the simple sine wave, and the simple sine wave is characterizing some of the light. This light is hitting a moving particle, and if the moving particle is reflecting in a certain angle, there is a change of the shape and the frequency of the light, which can be described the change in the amplitude and also change in the frequency. So therefore, if we use a sophisticated method, we can go from the time activity scale to the frequency scale. The monochromatic light, which had only one very narrow band, is widening, and the monochromatic light is transferred to a narrow, not as monochromatic as possible. So therefore, a laser is hitting the system, we collect reflected light, and from this reflected light we like to calculate the speed and the number of the moving particles within a certain volume. So it is again this principle. This is a superficial skin scheme. You can see also that the loops of the microcirculatory system are considered to be arranged randomly. Of course, there are very precious rules how this system is built up, but from optical point of view, there is a reflection from the surface, there's a penetration and reflection from everywhere, and we have to collect this reflected light and do something. We consider that the laser light had a single frequency and the reflected light had a wider band of frequencies, and we, with the Fourier transformation and other calculation processes, can calculate how much blood cells are in a certain volume and what is the speed distribution of them. In the practice, is it done in such a way? There is a system which is producing the laser light, and laser light is directed to the skin via optofibers. These optofibers are transporting in a safe way, and the optofibers are also good because there is not too much loss of the energy when it is transported from one place to another place. And of course, the reflected light could be also collected. In this case, there is no contact measurement. In this case, the laser light is projected to the skin surface, and the reflected light is picked up with a camera, and after it there is a computation procedure to color code and to see what's going on. If we just go back to the brain, this is a interesting picture, because someone filled a human brain with cast and digested out the rest of the brain. Only the blood vessels are visible. So you see that it is a very dense the system, and if you ask how is the blood flow in this complex system, it's very difficult to answer this question, because no direction, no proper velocity can be defined. So therefore, in a complex system, when we regard, either we measure the blood flow in big arteries, or we go into details and try to find out from a small area how is the entire brain working in this respect. So I started my research with lasers and the microcirculation 25 years ago, and did very simple measurements with single point lasers during the first years, and published some interesting papers about the dynamics of the cerebral microcirculation, because the laser technology enables a good temporal distribution, and a lot of dynamic properties were not described before the laser area. What is the advantage of the laser Doppler flow metric technique? It's very highly sensitive. Small change in velocity, small change in number of the blood cells could be checked. It is responsive to local blood perfusion, and enables us to make a real-time monitoring. Real-time monitoring is a big issue even now. We can now monitor live processes. I have a smart watch on my hand, and it is checking my blood pressure, and it can check also my pulse. We consider that more data we can have a better picture of the living system. It could be true or not, but in one way it is allowing us real-time monitoring, and a lot of aspects of the dynamics could be checked. It is non-invasive. This is a very crucial issue in the diagnosis, in the medical measurements. Less harm to the patient, less pain to the patient, less interaction between the living tissue and the measurement techniques. As I mentioned, now these low energy laser lights don't disturb the normal physiological state of the microcirculation. Small dimensions of the probes have enabled to be employed in experimental clinical environment. So not big measurement techniques are used. As I mentioned, in the skin microcirculation there are like contact optodes, so there are like EKG or other sensors, and we can heat or cool the regional area, and we can study local microcirculation. And also in one of the previous slides I just skipped it, there was a microperfusion system when some chemicals are perfused, either physically perfused or through iontophoresis, and change the microenvironment of the microvessels, and blood flow changes can be analyzed. So it says that a lot of knowledge gathered during the last 30 years regarding the vascular responsibility. What does it mean, vascular responsibility? We challenge the system somehow, for example, upload the forearm and release it, and how the answer is dynamically appearing. We can calculate on the nature of diseases. This is a typical measurement procedure. This is the normal microcirculation normalized to 0 to the basis. If the major arteries are occluded, there is a drop. Actually, there is no flow, and if it is released, there is a sudden increase in the blood flow, which is getting back to the normal way. This provocation, and the answer to the provocation is called reactive hyperemia, and a lot of data got available that these simple methods, or very simple methods, are indicative for renal diseases, for early stage of diabetes, for metabolic syndromes, and lot of changes which are also reflected in the skin microcirculation, but previously it was impossible to explore them because we didn't have appropriate measurement techniques. And this provocation is also continued by local heating, and you see that local heating also results in hyperemia. We know it well because we see that the local heating always results in change of color of the skin, and local heating results in a nice local hyperemia. And you see, this is not a linear, so there are people who are analyzing the rhythms appearing here, up and down, and this is also indicative, for example, for the status of the autonomic nervous system, how is it regulated by local and central factors. So we check the capillaries in the brain, we measure microcirculatory network, and this microcirculatory network is very complex. Lot of components are, these are the single layers of the endothelial cells, and the endothelial cells are accompanied with other cells which are preventing the free transport back and forth, and this system is also very complexly regulated. Of course, you can make a small window on the brain surface as well, and you can observe the blood vessels. And as I mentioned, it depends on the light you use as a light source. You can see reflected lights and absorbed light. It could be a basis of the advanced spectroscopy, but normally people observe the diameters and branching of this